Hello, class. Well, this is final review. We've done a game. Only 2 people joined, which is 1 including myself. So these results aren't the best, but we are going to use these results to go over questions that we found difficult or incorrect. We have these 9 questions that we all found incorrect, and we're going to look at these questions. We're going to assess these questions. We're going to find the right answers. We're going to look at the slide where they are at, and everything will be amazing. All right. Everything else we got 100 at. Just these 9 we struggled with. These 9. Me and Giselle with 1 L. We did this. We found 9 questions. We're going to go over these 9 questions for you guys. This is what we were gonna do on Wednesday. Hopefully we can do something similar on Monday and something on Thursday like this, because we do want to see what we need help on, what we're struggling in. So we're gonna go first thing. We're gonna go over pH levels. So I believe pH levels is in chapter 2. Chapter 2, chemistry of life. Chemistry of life. So we look at these. Let's wait for it to load. There's a lot of things that has to load at once. Okay, we got chemistry of life. So let's go back down to acid and bases. So the question we had was, your lemonade has a pH of 2. People don't like that. You can use a buffer such as water because it will accept H+. She's so nutrient. Let's look at what the slides say. So water molecules break into hydrogen plus and hydroxide, OH-. Acid releases H+. Bases accept H+. So the question is asking, we have a pH of 2. A pH of 2, is that acidic or base? Acidic or basic? So we have to look at this chart right here. Basic is 14. The closer you are to 14, the more basic you are. The closer you are to 0, the more acidic. 2 is closer to 0, so this is acidic. And here it says acidic things release hydrogen plus. A buffer minimizes changes in pH. Buffers can accept or donate H+. So this is what buffers do. They do the exact opposite of what it is. So the problem is it's releasing too much H+. It's too acidic, it's releasing too much H+. So we're going to use a buffer. We want to do the opposite, which is accepting. So let's look at our answer choices here. And we have release H+, neutralize with OH, and balance H plus with electrons, and accept H plus. Because it's acidic, we're going to release positive hydrogens. So the opposite will be accepting them. The second one is diffusion. Diffusion, I believe, is in 5. Lecture 5. Diffusion is highly important to understand. It might be a little confusing, but we're talking about passive transport. Passive transport does not require energy. This question is asking, in passive diffusion, molecules tend to move from an area of blank concentration to an area of blank concentration. So the right answer is higher to lower. So if we look at the slides, fusion happens at a higher concentration to lower concentration. Just look at these pictures. Higher transportation to lower transportation. Higher to lower. It automatically moves. It wants to reach stability. It wants to reach equal. Both sides want to be equal. So you'll have a lot of this and it'll automatically move so it can even out from high to low. So the next question we have. The next question we have is, what molecule donates electrons to the photosystem in light-dependent reactions? So we are going to go to our photosynthesis, right? And we have a video. Describing photosynthesis and cellular respiration. Please watch those. I'm gonna actually use the video instead to show. Here, we're in the perfect picture. This is the picture I was gonna use. So let me full screen this. So we have two photosystems. We have photosystem 2, photosystem 1. So what we're gonna use the light energy, which is photons, and transfer that into electrons. But how do we get the electrons? This is why plants need water, and this is why plants give out oxygen. Oxygen is given out because we don't need O2 from water. All we need is the hydrogen, because the hydrogen will donate the electrons, but we can't get hydrogen by itself. It's pretty rare on Earth, so instead we get water, we'll break it, and then 2 oxygen will leave the plant because it's a waste product, and then the hydrogen will donate its electrons. So please check out the videos. I highly recommend. Water is the correct answer, not ADP+. This one is 2, maybe 5. Okay, here it's in 5 and 2, maybe. Yes, 2. Same stuff. Okay. Solvent is the dissolving aid. Okay. Solvent is the water. In this example, the solvent. Okay, let me see. So we either have solution or solvent. I was wrong. It's solvent, but let's look at why. We're gonna use number 5. So we have a solution. A solution is a mix of a solvent and a solute. A solvent is what we're gonna use to dissolve. In this case, it's water. But in the test question, it was chocolate milk. And the solute is what's being dissolved. Let's say we're gonna dissolve salt. Water is highly common as a solvent because it's very good at dissolving things. Next question we have. It's been how long? Oh, it's only been 8 minutes, and we are in we're halfway here, but this next one describes if a chemical compound is an acid, it will. I got this one wrong. Accept H+ into the solution. So pH is kind of confusing. So let's go back to the pH. So pH releases and basic accepts. So if you look at the question and it asks if a chemical compound is an acid, we can already look here. Acids release hydrogen, not accept. What is not a factor that can influence the mixing of a molecule of a solute through liquid by diffusion? So let's go to our diffusion slides. And here we have concentration, because concentration is what really moves everything. So we can cross out. We can do a process of elimination. Concentration doesn't work because higher to lower, that's how the entire thing works. The next one we have is temperature. Temperature is, faster movement at higher temperature. We know molecules move faster when higher temperature. That's why when you heat something, it'll become a gas because the heat energy is making everything move faster. So temperature does make things go faster, so it's an effect. Size, you can look here. Size, small molecules move with less energy than large molecules. Smaller you are, the easier it is to move you than larger molecules. Common sense. Charge is another one. Opposite charge attract. A negative and a positive, they'll attract each other. And pressure, high pressure squeezes things together. These are the factors. But if we look here, the only factor that was here but not on that slide was aqueous solutions. So that would be the right one. Which organelle is responsible for modifying proteins and packing them into the vesicles? So this one, I would recommend looking at this. It's a little hard to see. I will show you the slides because so we have the endoplasmic rectum. It modifies and packs proteins. The ask is which organelle is responsible for modifying proteins and packing them into vesicles. That would be the endoplasmic rectum. The smooth breaks things down. The rough does the modification. It has ribosomes, so it's going to reduce proteins, modify these proteins, and pack them so it can be sent. I will show you the slides because I do not make it obvious in my poster thing. So I will show you the slides to help you. That poster will give you all what's in here in there. It could be a little messy, better, cleaner, but it's the best I could have done, basically, with the amount of time I had. We're gonna let it load a little bit. So while we let it load, we're gonna keep that. And we're gonna go. Which of the following conditions would result in the greatest amount of turgor that a plant cell possesses? So this one is hypotonic, not hypertonic. And we're going to look at the diffusion cells, of course. And right here, it says hypotonic turgor. So if you're in a hypotonic things will enter you because you are the low concentration. So the outside has to be the high concentration, which means things will enter you. And if too much stuff enters you, you might break. But that we're dealing with osmosis. So this is the difference. Even though the outside is hypotonic in solvents, I need to use vocab. Solutes, not solvents. Solutes, I'm sorry. Even though the outside might be lacking in solutes, it has more water. So the water will enter you while solutes will exit. And that will fill up your vacuole until because plants have a cell wall, it will try to keep everything out in place so you get this pressure inside. And that's what turgor is. Let's see if this has loaded. Yes. So let's talk about endorectum, rough endorectum, and okay, here. Okay, it says the rough is covered in ribosomes. Ribosomes make proteins, right? So let's go. Anything that deals with proteins, it's rough endoplasmic rectum. If it doesn't, it's smooth. So they lack ribosomes, but they synthesize lipids and detoxify enzymes. So because these have ribosomes, they can modify proteins and pack them. So we've done this and this. One more question. In a hypertonic solution, what is the direction of movement of the solutes? Remember, the solutes are things like salt molecules. They're not water. So if we look here, this stuff is confusing. Solutes. So if we're looking at solutes, it's asking in a hypertonic. So in a hypertonic, it would look like this. This is a hypertonic, and this is a hypotonic. This would be an example of hypo, but this is hyper. So there's more stuff outside. So stuff will enter your
Well, I love you. I love you so much. Wow. 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 I love you so much I love you.
So high concentration goes from outside to inside. We're done. If you have any other questions, please put those in the comments. I can help you. Very well. We're ready to help you. Please just ask your questions. And if any more questions come up, if more people play this Kahoot and get them wrong, I will explain them. Bye bye.